First of all, let's understand what is preeclampsia. Preeclampsia is basically a placental pathology characterized by new onset of high blood pressure along with proteinuria and or elevated liver enzymes that occurs after 20 weeks of gestation. Imprinted genes are known to play an important role in placental development, and they have found to be associated with preeclampsia. What are imprinted genes? Imprinted genes are those genes which are imprinted, that is methylated on any of the chromosomes, that is either maternal chromosome or the paternal chromosome. Likewise, if a gene is methylated or imprinted on the paternal chromosome, then its maternal allele only will get expressed and vice versa. DLK1 and pectin are paternally expressed imprinted genes, whereas PHLDA2 and CDKN1C are maternally expressed imprinted genes, and all these genes are known to play crucial roles in trophoblast biology and functions. There is a dangerous partner hypothesis also in preeclampsia, which states that there is a paternal risk factor involved in preeclampsia. Studies have shown that paternal genome is involved in placental development. Severe early preeclampsia is reported in diandric hydatidiform moles of paternal origin, and epidemiological studies say that in women with previous normal pregnancy, the change of partner increases the risk of preeclampsia. The lacuna that I observed was that no study has explored whether paternal epigenetic factors such as aberrant sperm DNA methylation could contribute to preeclampsia. The objectives are: number 1, to study the methylation of imprinted gene DMRs, DLK1, GTL2, IgDMR, KVDMR, and pectin DMR in the spermatozoa of preeclampsia cases. And the second objective is to study the expression of these imprinted genes in the placentae of preeclampsia cases. The methodology is: semen and placental villi samples are being collected, the sperm DNA is extracted, subjected to bisulfite modification and pyrosequencing. From the placental villi, RNA is extracted and subjected to qPCR. Clinical history entails that in the control group, the gestational age at delivery was higher as compared to the case group. This is owing to the premature delivery of babies in the preeclampsia, the case group. The placental weight and the birth weight were also significantly lesser in the case group. In the case group, there were 3 instances of intrauterine fetal death and 1 case of intrauterine growth restriction, indicating that preeclampsia poses significant morbidity and mortality. The results of the objective 1 is that KVDMR and pectin DMR were found to be hypermethylated in the spermatozoa of the preeclampsia group, whereas IgDMR was found to be hypomethylated. In these 3 DMRs, 9 CpGs, 5 CpGs, and 7 CpGs have been studied respectively. In the second objective about gene expression, the PHLDA2 and CDKN1C and DLK1 expression was found to be upregulated in the villi of preeclampsia cases. The pectin expression did not come significant. So, in the present study, the hypermethylation of KVDMR in the spermatozoa of cases translated to elevated expression of imprinted genes in the placentae of the cases, indicating that methylation of this DMR is crucial for proper expression of the corresponding genes. And pectin DMR was found to be hypermethylated, but its expression was not abnormal in the placentae. The IgDMR showed hypomethylation in the sperm of the cases, but its expression was upregulated in the placentae, indicating that some compensatory mechanisms are at play. The conclusion I have represented in a graphical form here. I will go to the highlights. That is, the epigenetic contribution of the paternal genome to preeclampsia till now has not been reported earlier. The imprinted genes that were known to play a crucial role in placental development have been taken for studying in this research project. The methylation status of the imprinted genes in the spermatozoa has not been reported, which this study aims to report. And pyrosequencing has been used as a technique to study the CpG site-specific methylation. The clinical implications of the project are development of preconceptional methylation diagnostic marker for preeclampsia with sperm as a sample, and the way forward is to establish threshold methylation levels of imprinted genes for preeclampsia in the sperm DNA. Thank you for your patient hearing.